Okay, time to talk about transcription and translation. Just taking a look at this graphic here, this is a eukaryotic cell, probably an animal cell, and as is characteristic of all eukaryotic cells, we have our membrane-bound nucleus, and then of course inside that nucleus is our DNA. Now remember that the DNA is a very large molecule, and it actually is in fact too big to actually pass through the pores that are in the nuclear membrane, and as a result we need a way to get that genetic information out of the nucleus and into the cytoplasm, because that's where the ribosomes are, and the ribosomes of course are necessary for that protein synthesis. So essentially what we have to solve that problem in eukaryotic organisms is we have something called mRNA, which stands for messenger RNA. RNA is single-stranded and mRNA is quite small. The molecule actually is small enough that once it's formed, it can leave the nucleus, just like it shows in the graphic here, go into the cytoplasm, and then engage with the ribosome in order to make protein synthesis. So that process that happens inside the nucleus of creating that mRNA strand off of that DNA strand template, that is called transcription. Okay, so essentially the mRNA is taking that information, transcribing that information from the DNA molecule in a small enough form that it can be carried into the cytoplasm where the ribosome is, and then the next process, which is translation, can happen. So if we take a look at the actual process in terms of a little more detail, mRNA copies and transfers information from the DNA molecule inside the nucleus, and then we'll take that to the cytoplasm. So just like we had with DNA replication, before transcription happens, the chains of DNA are going to partially unwind, and they're going to expose part of that genetic code. And as that double helix uncoils, mRNA nucleotides, remember those are like the phosphate, the ribose sugar, etc., floating around in the nucleus, are going to bind to those DNA bases that have now been exposed and are looking for another nitrogenous base to bind to. An mRNA molecule forms as the nucleotides match, so those base pairs actually match up on that exposed DNA template. And then once it forms that chain of necessary genetic information and copies it, then it's going to carry it into the nucleus and into the cytoplasm in search of that ribosome, so that the next process, translation, can happen. Once that mRNA has left the nucleus, that DNA strand is going to recoil back into that nice double helix. Just like in DNA replication, we had DNA polymerase always working in the 5' to 3' direction. RNA polymerase, which is the same type of enzymes, except it works on RNA, works in the 5' to 3' direction as well. So if we take a look at our graphic here, this is essentially the process of transcription. So remember where we are. We are in the nucleus of a eukaryotic organism, and the purpose of this particular process is to get that genetic information from the DNA in order to code for the specific protein required by a cell, but in a small enough form that it can fit out of that nuclear membrane and into the cytoplasm. So here we have our DNA strand, and then what eventually happens before transcription takes place is that DNA is going to uncoil, and then you'll notice here that it has kind of this exposed, right? And you can tell that this is the DNA part, right? Because it has the T's, the C's, and the A's, etc. And so that part of the DNA strand is exposed. And so now what happens is this big enzyme here, which is RNA polymerase, so this is the builder of transcription, just like DNA polymerase was the builder of DNA replication. So we have that RNA polymerase moving along, and what that polymerase does is it grabs those nucleotides that are floating around in the nucleus, and it matches them up to that exposed DNA template strand. Okay, so the template is always what we're working off of. Now you'll notice though with the mRNA, you'll notice that of course we have our A and our T, but remember, because this is our RNA strand here, you'll notice that we have uracil on the RNA strand instead of adenine. So, or instead of thymine, I should say. So on our DNA strand, you'll notice that we have our regular 4 nitrogenous bases, right? Our thymine, our adenine, our guanine, our cytosine. But because the mRNA is RNA, anywhere on the DNA template strand that we have an adenine, we're going to have a uracil instead. Okay, once the transcription is done, so once we have this nice little piece of newly made mRNA that has grabbed that information, copied that information from the DNA strand. What ends up happening then is it separates, moves out of the nucleus and into the cytoplasm to look for a ribosome, and then the DNA will recoil itself back up. So translation is the second piece, okay? So transcription has taken place, the mRNA is now in the nucleus in the cytoplasm, and now we're going to get ready for the translation process, which is actually taking that information from the mRNA strand that left the nucleus and went into the cytoplasm, and then using that ribosome to actually make a protein. So the first part is what we call initiation. So the ribosome will bind to the mRNA, and it will move along the strand and read it, starting with that start or initiator codon, AUG. So remember we talked about on the mRNA chart, AUG is our initiator or our start codon. So our large subunit and our small subunit of our ribosome attach around that mRNA strand and basically reads it like a little tape running through that large and that small subunit, starting with that AUG or starter codon, and that's why it's called initiation, because it's the beginning of the formation of the amino acid chain. Each time the ribosome reads a new codon, and remember our triplet rule, a codon always has the 3 nitrogenous bases. Every time it reads a new codon, it's going to add another amino acid to that chain, and then of course as it grows, we call it a polypeptide chain, right? So many peptide bonds. The next RNA to become involved here is what we call the transfer RNA. So remember, the whole point of the messenger RNA is to get the information from the DNA out of the nucleus into the cytoplasm, because the DNA is too large to get out. And now the whole purpose of the tRNA, the transfer RNA, is to get the appropriate amino acid from the cytoplasm to match that mRNA strand so that we can create a polypeptide chain. So this is the essential graphic of what a tRNA molecule looks like. And so it kind of looks like this odd sort of puzzle-shaped piece. But the thing that we really need to kind of key in on here is at the bottom, we have a codon, okay? And please notice this is an mRNA codon, okay? And then at the top here, we see that the tRNA molecule is bringing a specific amino acid. So floating around in the cytoplasm, you have all of these amino acids that are kind of just waiting to get pulled in to a polypeptide chain and potentially become part of a protein molecule. So there's many different tRNA molecules, and each one has a specific what we call anticodon. That's a very, very important term. Okay, and kids quite often get the codon and the anticodon confused. Now I want you to think of what that prefix anti means. It means like in opposition to or opposite of, right, or opposing to. So an anticodon is a group of 3 complementary bases on the tRNA that recognize and pair with a codon on the mRNA. So when we talk about a codon, that's always messenger RNA. That's our triplet, that's on our little chart, okay? That's what codes for the amino acid. The anticodon, I want you to always associate with the transfer RNA. And the whole point of that is once the anticodon and the codon match up with one another, the amino acid is released and becomes part of that polypeptide chain. So each tRNA is able to carry an appropriate amino acid that the mRNA codes for to the ribosome, and then the ribosome links those into that polypeptide chain. So here's a graphic of what we were just talking about with the polypeptide chain synthesis. So here we have our ribosome, and you'll notice that our large and our small subunits of the ribosome are engaged with one another because we have a piece of mRNA here. So because we have our mRNA, our messenger RNA, we have the instructions from the DNA, okay? The mRNA formed in the nucleus, brought that information from the nucleus to the cytoplasm and engaged with the large and the small subunit of the ribosome. So one thing I want you to notice here along this green part here, this is our messenger RNA. Okay, so we have U, G, G, A, etc. Now these little puzzle pieces here, this is our tRNA, and I want you to notice that when our tRNA engages with our mRNA, we have the matching codon. Okay, so U goes with A on the mRNA, U goes with A, U goes with A, C goes with G, U goes with A goes with U, etc. So you'll notice that the anticodon, okay, is the one that is complementary to the mRNA strand. But what is really key to remember here is that it is the code on the mRNA strand that tells the tRNA which amino acid to bring. So AAA codes for lysine. So UUU on our tRNA strand does not code for lysine. And that's the biggest mistake kids make, is they look at the tRNA and they go, oh, UUU, that's going to make this amino acid. That's wrong. It's always the mRNA that you look at to decide which amino acid is going to be connected here. GAU codes for asparine. UUC codes for phenylalanine. Okay, so you never want to look at your codon chart in your data booklet and go from your tRNA. Your data booklet, that codon chart is mRNA, and that's the one that you have to look at, okay, in order to decide which amino acids are being put together in that polypeptide chain. The next part is elongation, and that just means that the ribosome, after initiation, continues to add on amino acids, add on amino acids, add on amino acids, until the entire mRNA strand is read and the whole polypeptide chain is formed. Okay, so that's kind of common sense to me. Initiation, ribosome engages. We have that starter codon, the methionine, the AUG, and the reading of the mRNA strand begins. Elongation means that the ribosome and the tRNA are working to bring the appropriate amino acids to make that polypeptide chain longer, right? So that's the elongation part, and forming that polypeptide. And then the last thing, as the name implies, is termination. And of course, termination, exactly as the name implies, eventually the ribosome is reading those codons, reading those codons, reading those codons, and then eventually it gets to one of those stop codons. So remember, on your codon chart, you have 3 stop codons, UGA, UAG, or UAA. So as soon as the ribosome reads one of those, that is a stop codon because it does not code for an amino acid. And then what ends up happening is the polypeptide chain is terminated and released, because those 3 codons, unlike AUG, the starter codon, that codes for methionine, okay, and that's the starter codon for all eukaryotic organisms. However, those stop codons, or UGA, etc., those do not code for an amino acid. So what effectively happens is the polypeptide chain is terminated, the ribosome falls off, okay, so that large subunit, small subunit separates and falls off from each other, and the polypeptide chain is released. And once that happens, then we can have the formation of our protein. So the polypeptide chain is the linear form, that primary, then it will spiral or pleat, our secondary, and so on, until it folds into a complete functional protein. So this is just a summary of kind of what's happening where. So once again, remember that the transcription piece is happening in the nucleus, okay? So this is our DNA template strand. We know this is DNA because it doesn't have uracil, okay? And then in the nucleus, RNA polymerase is going to form this mRNA strand in the 5' to 3' direction, okay, so it's building it this way in the 5' to 3' direction, and we know that this is RNA because it has the uracil, right? And each one of these triplets is a codon for a specific amino acid, okay, so all of that is happening in the nucleus. Now, once that mRNA strand is formed, it's going to leave the nucleus and it's going to go into the cytoplasm, and it's going to engage with a ribosome, and then that process is translation. So the ribosome reading that mRNA strand, the tRNA bringing the appropriate anticodons and with them the appropriate amino acid. we get the formation of that polypeptide chain, and then once we hit a stop codon, it's released and eventually will form into a protein, as it folds into that quaternary form. Now again, key here, kiddos, is it's the mRNA strand, not the tRNA strand, that codes for the amino acid. So GUU codes for valine, GCC codes for alanine, and so on. So that's the key component with translation, is the tRNA brings the anticodon and then brings with it that appropriate amino acid to form that polypeptide chain.